Welcome to Biotanemy.
Signal transduction pathways. Signal transduction is the process by which a cell converts an extracellular signal, like a hormone, growth factor, or stress signal, into a specific intracellular response, ultimately affecting gene expression, metabolism, cell survival, or proliferation. The key features of the signal transduction pathway are 1. Signal reception. 2. Signal transduction. 3. Signal amplification. 4. Signal integration. 5. Cellular response. And components of signal transduction are 1. Ligand. 2. Receptor. 3. Second messenger. 4. Effector proteins. 5. Adaptor proteins. Based on the types of receptors, the signal transduction is divided into 5. 1. G-protein coupled receptors or GPCRs. GPCRs are 7 transmembrane domain receptors embedded in the plasma membrane. They detect extracellular ligands like hormones, neurotransmitters, or sensory stimuli. The ligand binds to the extracellular part of the GPCR. This causes a conformational change in the GPCR. The conformational change allows the GPCR to act as a guanine nucleotide exchange factor, which leads to the replacement of GDP on the G-alpha subunit by GTP, activating G-alpha. The G-alpha GTP subunit dissociates from the G-beta gamma subunits. G-alpha GTP will activate downstream effector proteins like adenylyl cyclase and phospholipase C, while G-beta gamma will activate ion channels leading to changes in membrane potential or ion flux. Activation of kinases, ion channels, or transcription factors leads to specific cellular responses, such as metabolism changes, secretion, and gene expression changes. The signals are terminated by GTP hydrolysis on G-alpha. Ligand dissociates from GPCR. Beta-arrestins can desensitize or internalize the receptor. Second messengers are degraded or sequestered. 2. Receptor tyrosine kinases. RTKs are single-pass transmembrane receptors with extracellular domain that binds ligands such as growth factors, for example, EGFR, VEGFR, and insulin receptor, while the intracellular tyrosine kinase domain will catalyze phosphorylation. The binding of ligand to the extracellular domain of the RTK will trigger dimerisation. Dimerisation activates the kinase domain, which phosphorylates tyrosine residues on the receptor itself. These phosphotyrosines act as docking sites for downstream signaling proteins with SH2 or PTB domains. The adaptor proteins and effectors that are activated by SH2 are 1. GRB2 binds to phosphotyrosines to recruit SOS and activate RAS. 2. PI3K binds specific phosphotyrosines to converts PIP2 to PIP3 and activates AKT. 3. PLC gamma activation leads to the generation of IP3 and DAG to activates PKC and CA superscript 2 signaling. RTKs can influence the downstream signaling cascades, like MAPK or ERK pathway leads to gene expression for proliferation, differentiation. PI3K or AKT pathway that promotes cell survival, growth, and metabolism. JAK or STAT pathway will lead to transcription of target genes. The signal is terminated by receptor internalization and degradation. Dephosphorylation by tyrosine phosphatases. Negative feedback loops in downstream pathways. 3. Cytokine receptors. Cytokine receptors do not have intrinsic kinase activity. They are single-pass transmembrane receptors that associate with cytoplasmic kinases, mainly Janus kinases or JAKs. Examples: interleukin receptors, interferon receptors. A cytokine binds to the extracellular domain of its receptor. This induces dimerisation or a conformational change in the receptor. Each receptor subunit is associated with a JAK. Dimerisation brings JAKs close together. They phosphorylate each other and the receptor on specific tyrosine residues. Phosphorylated tyrosines on the receptor act as docking sites for STATs via their SH2 domains.
STAT dimers translocate to the nucleus, bind DNA, and regulate transcription of target genes. For example, JAK-STAT in immune response, cell growth, and survival. The signal is terminated by 1. Dephosphorylation of JAKs, STATs, and receptor tyrosines by phosphatases. 2. SOCS proteins inhibit JAK activity and promote receptor degradation. 4. Ion channel-linked receptors. These receptors are transmembrane proteins that form a pore or ion channel in the membrane. They open or close in response to ligand binding. Common examples are the nicotinic acetylcholine receptor, GABA receptor, and NMDA receptor. The ligand, such as acetylcholine, glutamate, and GABA, binds to the extracellular domain of the receptor. This causes a conformational change in the receptor. The conformational change opens the ion channel, allowing specific ions such as sodium, potassium, calcium, and chloride to flow across the membrane. Rapid influx or efflux of ions changes the membrane potential, leading to depolarization or excitatory response and hyperpolarization or inhibitory response. Changes in membrane potential trigger physiological responses such as neurotransmission in neurons, muscle contraction in skeletal or cardiac muscle, and secretion in endocrine or exocrine cells. The signals are terminated through ligand unbinding, leading to the closure of the channel and the restoration of ion gradients by pumps such as sodium-potassium ADPase and calcium pumps. Rapid response ensures fast signaling. 5. Intracellular or nuclear receptors. These receptors are located inside the cell, in the cytoplasm or nucleus, not on the cell surface. They act as ligand-activated transcription factors. Examples are glucocorticoid receptor, estrogen receptor, androgen receptor, thyroid hormone receptor, retinoic acid receptor. The ligands seen in here are small, lipophilic molecules that can easily diffuse through the plasma membrane. For example, steroid hormones, thyroid hormones, vitamin D, and retinoic acid. The hormone or signaling molecule diffuses across the cell membrane. The ligand binds to its specific receptor in the cytoplasm or nucleus. This causes a conformational change in the receptor. Inactive receptors are often bound to inhibitory proteins, like HSP90. Ligand binding releases these inhibitors, activating the receptor. Activated receptors dimerize to form homo or heterodimers. The receptor complex then binds to specific DNA sequences called hormone response elements in target gene promoters. The receptor DNA complex recruits coactivators or co-repressors to regulate RNA polymerase II activity. This results in activation or repression of target gene transcription. The expressed proteins control various biological processes, such as metabolism through glucocorticoids, reproduction and development, with help of estrogen and androgen. Also participate in homeostasis and differentiation. The signals are terminated through hormone degradation or dissociation, receptor downregulation or export from the nucleus, and feedback inhibition at the transcriptional level.
So, now you guys have a basic understanding of the components of signal transduction. Let's see about the major common signal transduction pathways in the next video.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.